Hi, this lecture is about enzymes. So hopefully you just watched the lecture on proteins, and within that lecture I mentioned enzymes. Enzymes are a group of biological catalysts, meaning they're things that we find in living systems and they speed up chemical reactions. So enzymes are one of the most important groups of proteins. I have a little star by that. So it turns out that most enzymes, but not all enzymes, are made out of protein. There are a few enzymes that are made out of RNA, but 99.9% of all enzymes are made out of protein. So I usually talk about enzymes right after I talk about proteins. Now, recall that for life to occur, chemical reactions must occur. One of the properties of living things is something called metabolism, which is really just a sum of chemical reactions. And these chemical reactions are bringing about the processes of life. It turns out that these chemical reactions that sustain life, they really can't occur fast enough without the presence of enzymes. So enzymes basically speed up chemical reactions to the point where life can actually occur. So we say that enzymes catalyze chemical reactions. And there's different ways of thinking about this. Some people say that this means that enzymes increase the rate of a chemical reaction. Some say that it speeds up a chemical reaction. Some people interpret this as it makes it easier for chemical reactions to occur. In a way, all of these are correct statements. On screen, there we have a picture of a chemical reaction. So we have a molecule called sucrose, which is also known as table sugar, okay? And it's being broken down into its two components, glucose and fructose, right? So sucrose is a disaccharide, and after this chemical reaction is complete, we have two monosaccharides, glucose and fructose. Okay? In order for this reaction to occur at a rate that is compatible with life, an enzyme is required. If we want to break down sugar inside of our cells, and we want to do it fast enough that we can actually live, we need an enzyme to help this process along. Okay? The way that an enzyme is able to do this is an enzyme lowers the startup energy for a chemical reaction. So every chemical reaction requires a little bit of startup energy to get it going, and enzymes lower that startup energy. The startup energy for a chemical reaction is called activation energy. Right? So enzymes lower the activation energy for a reaction. By lowering the activation energy, this basically means it's going to be easier and faster for the reaction to start. Normally, I would graph a catalyzed and an uncatalyzed normally I would graph an uncatalyzed reaction and then graph alongside that a catalyzed enzymatic reaction. But because I'm using this PowerPoint lecture, I'm going to just show you a picture. This is pretty much the same thing that I would draw anyway. So let me kind of explain this graph. Okay, going up over here, we have energy. So up at the top, very high energy, down at the bottom, low energy. Okay. Along the bottom, we have reaction progress, and what this is saying is that the reaction is starting over here, and the reaction ends way over here. Okay. We have two lines on this graph. We have this black line, which is representing an uncatalyzed reaction. So that's a reaction with no enzyme. And then in red, we have the catalyzed reaction. Right? So this would be the reaction in the presence of an enzyme. So first you should notice that with these two reactions, they both start at the same energy level, okay? And they both end at the same energy level. So they're the same in terms of their energy level in the beginning and the end, okay? The big difference is here in the middle, right? This double-sided arrow here, this is the activation energy for this reaction when there's no enzyme present. The red double-sided arrow is the activation energy in the presence of an enzyme. So basically what this is saying is that in order for this reaction to occur, we have this big hump of energy that we have to get over first. Right? When there's no enzyme, this is a really big hump of energy we have to get past in order for this reaction to proceed. But if we have an enzyme present, it's just a little bump of energy that we have to get past. So you can see how the enzyme would make it a lot easier for this reaction to occur. The enzyme has lowered the activation energy for this reaction. This is sort of a different way to kind of represent the same thing. So we have something over here and a wall, and it's jumping over the wall, and once it jumps over the wall, it splits into two things, right? So essentially this is just another way to represent a chemical reaction, right? This is our starting product, right? Actually, I shouldn't say starting product, that's sort of misleading. This is the reactant, I should say, and over here, these are the products that we get at the end. Okay? So you can think of this as like sucrose, okay? Sucrose, and it's going to be broken down into glucose and fructose, right? So take a minute and see if you can decide what number 3 is representing. What is this wall representing here? And if you said activation energy, that's correct. So there's a wall of energy that we have to get over in order for this reaction to proceed. And so what an enzyme would do is it would make this wall shorter. It'd be easier for the reaction to jump over that wall if it was shorter. So how exactly do enzymes lower activation energy? I use a little analogy to kind of describe this. Think of an enzyme as a stage on which a chemical reaction can occur. And so the chemical reaction is sort of like a play, and this play is happening on a stage, the stage being the enzyme. So the enzyme is just giving a place for the reaction to occur. Right? Basically, the enzyme makes sure that all the actors are present and standing in just the right spot so that the play can go on. Right? In this analogy, the actors are the substrates, or the reactants, if you will. So that's kind of what you start off with, the before things. Right? Again, the stage is the enzyme and the play is the reaction. In this analogy, the actor has to stand in just one spot on the stage, right? You know, obviously when we go see a play, actors move around on the stage, but in this particular analogy, the actors have to stand in just one spot on the stage, and that spot is called the active site. So the active site is actually a part on the enzyme where a substrate will fit. And this picture is here just to show you that the active sites on an enzyme are very specific to their substrates. Right? So here we have a generic enzyme, enzyme X, and you can see this is its active site. And if you look at all these various substrates, you can see only certain substrates are going to fit into this active site. Right? The square peg is not going to fit into the round hole. Right? So again, over here looking at this different enzyme, you can see it has a very specific shape to its active site, and only certain substrates will fit. They say that this is very much analogous to a lock and a key, right? So a key only fits a certain lock. So certain substrates can only fit into certain enzymes. So enzymes can catalyze both catabolic and anabolic reactions. Anabolic reactions are reactions that are used to build molecules, while catabolic reactions are the reactions that are used to break molecules down. And enzymes are used in both circumstances. So let's look first at how an enzyme is used during an anabolic reaction. Okay, so we're going to use an enzyme to help build a molecule. Right. So you can see here we're starting off with two pieces, two reactants, reactant 1, reactant 2, or what we call substrates in the case of a enzyme catalyzed reaction. So we got substrate 1 and substrate 2, and we're going to end up with just one full product at the end. So what's going to happen is substrate 1 and substrate 2 is going to come together and form a bond and become one. Okay? And let's look at how the enzyme can be involved with this process. Okay? So here we have our enzyme, our stage, right? And there's our active sites, the part on the stage where these substrates will fit. Right? If you notice, this substrate is only going to fit into the enzyme in one specific orientation. It can't flip upside down and come in. It can't flip sideways and come in. Right? It can only fit such that this little knobby area will fit into the active site. And the same thing goes for substrate number 2. Right? Substrate number 2 can't come in upside down or sideways. It has to come in such a way that this little area will fit into the active site. That's the only way the two will ever come to the enzyme. Right? And if you notice, when the two substrates come into the enzyme, these little pink areas are now next to each other. You can see the pink areas here and here when they're separate, right? But when they come and sit on the enzyme, those little pink areas are next to each other. And those pink areas are going to contain functional groups, so groups that could react with each other, and indeed they do. If they come close enough together, a reaction will occur, forming a bond between substrate 1 and substrate 2, forming a single product that can then be released by the enzyme. So basically, the enzyme is just ensuring that the substrates are perfectly positioned in such a way that their functional groups can come close together and react and form the bond. Okay? So really, if you notice, the enzyme is not changing in this whole entire process, right? It can be used again and again. Right? The enzyme didn't supply any energy to the reaction. It was literally just a place for the substrates to come into. Now let's look at how an enzyme can be used during a catabolic reaction. So we're going to use an enzyme to break something down. Right? So here we have our substrate that we're going to break down into two separate pieces. You can see at the end it's going to look like this. Okay? So here is our enzyme and the active site. So the substrate will come in and bind to the active site on the enzyme. Right? Now sometimes when a substrate comes and binds to an enzyme, once the two fit together, the enzyme might squeeze on the substrate a little bit. I kind of think of this as the equivalent of the floorboards on the stage kind of creaking and bending a little bit when someone stands on a stage. So there's a little bend, a little give on the enzyme when the substrate comes and binds. And when this happens, sometimes the enzyme will just pull a little bit on the substrate, adding stress to some bonds within the substrate. So basically what's happening here is once the substrate comes and binds to the enzyme's going to just change shape a little bit, just like the floorboards creaking, and we're going to put some stress on this bond when that happens. And if a drop of water should come in, that's all we need, just a drop of water to come in and break that stressed out bond. And now we have our two separate products that can be released. So basically what we're seeing here is upon binding, the substrate is slightly bent, placing stress on some of the bonds, making it a lot easier for those bonds to break. Right? The stressed bonds often become more accessible to water, which can be used to break the bonds. So here we've used an enzyme to break a molecule down. We've used an enzyme in a catabolic reaction. The enzyme can be used again and again. The enzyme is just a stage on which this reaction occurred. The enzyme did not add any energy to this reaction, but what it did do is it lowered the activation energy and made it easier for this reaction to occur. I'm going to bring you back a slide because I wanted to say something about this, but I couldn't remember what it was I wanted to say, and I moved on to the next slide, so I remembered what I wanted to tell you here. Actually, it's a question. I want to ask you, what do you think the chances are that substrate 2 and substrate 1 would bump into each other at just the right orientation, such that these little pink areas would be right next to each other? What are the chances that that would happen without the enzyme? What are the chances that those two things would come together at just the perfect orientation so that a bond could form between them? And if you said that the chances are very low that that could happen, you're right. And this is where an enzyme comes in. An enzyme is basically providing an area for this reaction to occur. It's making sure that these substrates come together at the right angle so that the reaction can occur. So here we see an enzyme and a substrate, and here's the enzyme's active site. Here's what it looks like when the enzyme is bound to its substrate. So my question to you is, what's happening in these two pictures? See if you can figure it out. You can just think about this in general terms. You don't have to use any fancy techie terms. So go ahead and pause the video right now and see if you can come up with a description of what's going on here. So maybe for this one you said that, okay, there's something coming in that's not the substrate, that's blocking the active site so that the substrate can't bind. And absolutely, that is what's happening here. Now over here we have kind of the same thing happening but in a different way. We have something coming in and binding to the enzyme, but it's not binding at the active site, but it's causing a change, a shape change in the enzyme. See how it made the active site smaller, and now the substrate can't bind. So effectively, the same thing has happened in both of these, in that the substrate can't bind. And what we're looking at here are examples of something called inhibition. we have things coming into the enzyme inhibiting it from working. And that's one way that enzymes are regulated, is by things called inhibitors. So enzymes aren't always constantly turned on. They are turned off by things like inhibitors. So ultimately, the take-home message here is that enzyme activity can be regulated. Take a minute to look at this graph and see if you can answer the questions here. So go ahead and pause the video, look at the graph, and take a minute to see if you can answer the questions. Alright, so on this graph we have a graph for enzyme A and a graph for enzyme B. We have temperature down here and rate of reaction over here. So the question says, at what temperature does enzyme A work best? Right? So the peak of the reaction rate, the highest reaction rate for enzyme A is right here, and it's at around like 38 degrees Celsius. So enzyme A works best at 38 degrees Celsius. That's called the optimal temperature. So every enzyme has an optimal temperature at which it works best. The next question says, what happens to reaction rate if you go above the optimal temperature? Well, let's look at the case for enzyme A. Let's say we go up to 45 degrees Celsius. Well, then reaction rate decreases, doesn't it? So the optimal temperature is the temperature at which the enzyme works best. If you go below that or above that, the enzyme is not going to work as well. Again, pause the video at this point, take a look at the graph, and read the questions and see if you can answer the questions. So in this particular graph, we still have rate of reaction on this side, but down here we have pH. Okay? We have two enzymes in question, pepsin and trypsin. First question says, what enzyme operates best at pH of 6? So we come over here, we find pH 6, and we see that, well, pepsin doesn't work at all at pH 6, but trypsin does, right? What is the optimal pH of the enzyme pepsin? So we want to come to the peak of the rate of reaction for pepsin, and we see that it works best at around 2.5 in terms of pH. So it works best at a pH of about 2.5, which is pretty acidic, actually. Why doesn't trypsin work well at pH 3? Right? Well, as with temperature, an enzyme has an optimal pH at which it works best. Trypsin works best at a pH of about 6.5. If you go above or below that, actually what's happening here is the trypsin is denaturing, it's unfolding. So in a very acidic environment, such as a pH 3, the trypsin is completely unfolded or completely denatured, and it won't work. So some enzyme characteristics. Enzymes are specific, meaning that one enzyme catalyzes usually only one specific reaction, though oftentimes the reaction can be reversible. So what this is saying is that an enzyme can work in both the forward and reverse reaction. For example, the enzyme sucrase can break down sucrose, forming glucose and fructose, but it can actually work in the reverse direction too. Sucrase can be used to combine glucose and fructose to form sucrose. Enzymes can be regulated. They can be turned off and turned on. An example of that was inhibition that we looked at. Enzymes can be reused again and again. And an enzyme works best at their optimal temperature and at their optimal pH.